Good evening, everybody. Can everybody hear me okay at the back? So as it's a very kind opportunity to be able to speak to you this afternoon on this very special day, International Epilepsy Day. And as Peter said, I'm the Professor of Neurophysiology of Epilepsy. My full title is actually the Ellen Mayston Bates Professor of Neurophysiology of Epilepsy. And as you look at this slide tonight, there's probably lots of questions that are running through your mind. What is neurophysiology? Who is Ellen Mayston Bates? And what I'm going to try and do is actually to set the scene by answering some of these questions, but that will sort of feed into the main part of my presentation this evening. So who was Ellen Mayston Bates? Well, this is a picture of Ellen Mayston Bates, and through her very generous bequeathment, my chair and a soon-to-be appointed clinical chair in epileptology has been very generously appointed at Trinity College Dublin. We know very little about Ellen Mayston Bates. Obviously, she made this very generous bequeathment for research into epilepsy to take place at Trinity College Dublin. She passed away in 2009. She's a bit of a mystery. What we do know is that a family member was thought to have epilepsy, and she obviously was moved to make this bequest to try and fund research into this debilitating neurological condition. I think the really important message that I'd like to get across with this slide is the power of giving. And, you know, this lady has obviously given so much for epilepsy research here in Ireland. But I think as a research community, it's also incumbent upon us to give something back. And obviously, an event like tonight allows the research community to do that through education, through telling you about the work that we do, but also hopefully through engagement, which we've heard about, and through advocacy, and through our research outputs. And so with research in mind, I'd like to share some of our research that we've done. And in doing that, I'd like to then address one of the other questions which arose from my first slide, which is, what is neurophysiology? So if you go to the Oxford English Dictionary, it's a noun, and it means the study of the functions of the nervous system. And, of course, the next question is one that you're probably all familiar with. What is epilepsy? Well, you know that. It's a neurological condition. It's characterized by recurrent seizures, and these seizures are due to brief disturbances, brief electrical disturbances in the electrical functions of the brain. And neurophysiology really is the ability by which we try and study and understand how these disturbances in the electrical connectivity and the chemical transmission between brain cells give rise to this aberrant activity. And of course, epilepsy is a disease which can affect across the lifespan. And we know that the causes are multiple, from CNS infection, head injury, genetic basis, which we've heard a lot about, perinatal or prenatal brain damage, brain tumours, ageing, and unknown causes. But at a fundamental level, these disturbances in electrical activity are at the very core of those seizures that are manifest in epilepsy. Now, I think it's quite timely that epilepsy is really sort of at the forefront of research here in Ireland, because there's actually strong historical precedents for this. This gentleman here is Robert Todd. He may be familiar to the neurologists in the audience. Todd was actually born in Dublin. He, back in the 19th century, actually proposed an electrical theory of epilepsy. And his legacy to epileptology and to neurosciences is invaluable. And in fact, the electrical basis of epilepsy, which he proposed, was confirmed by the EEG, the electroencephalogram recordings that were discovered by Hans Berger in the early 20th century. So I think it's sort of timely, and it makes sense that Dublin is at the very heart of epilepsy research in Europe. And the important thing for researchers is obviously we need to be able to study the neurophysiology of epilepsy. And what is unique is that we can do this at multiple levels. So we can study the neurophysiology of epilepsy at the level of the whole brain in patients by doing EEG recordings. And we see this type of sort of seizure-like activity. So this is an electrical recording of a seizure-like event. But what is important for us as researchers is that we can look at this at different levels and across different species. So we can study it in the lab. We can use rats or mice, and we can record from their brain as they move around and are having seizures. And we do this using a technique called ECOG, electrocardiogram. And again, the patterns of activity are strikingly similar. So this electrical activity is conserved across species. And we can even produce brain slices. So we can take the brain from this animal, we can cut it into very thin slices, and we can study critical parts of the brain. For example, as shown here, the hippocampus. And we can essentially study seizure activities in these slices using local field potentials. And it is these local field potentials which essentially underlie the patterns of activity that are recorded in the EEG. So there's a direct translational link between the recordings that we do at the level of brain slices right up to the levels of recordings that we do at the level of the whole brain. And in fact, we can even now study epilepsy using computational models. So we can use very powerful in silico or computer chips to essentially recapitulate the seizure-like activity as well. So there's different ways in which we can study this electrical activity. And I'm now going to try and show you some of the examples from my own work about how we've used these techniques to study epilepsy, and hopefully also demonstrate how we can translate that work from what we do in the lab to the bedside. So how do we study epilepsy? Well, this is just a brain scan which shows the hippocampus here in a patient with temporal lobe epilepsy. And we use acute and chronic animal models, as we've already heard of before, to study epilepsy. I suppose the unique thing that we do is we use human tissue obtained from patients like this with a history of seizures of epilepsy. And we can actually obtain the surgical tissue when it's removed, obviously with consent from the patient. We can keep that tissue alive, and we can record from the brain cells in those slices of human tissue and record the seizure activity that is occurring. So this gets us a step closer to the patient, and we use these electrophysiological tools to do that, which I'll tell you a little bit more about. We use pharmacological tools, so we can interrogate whether we can block or activate certain receptors or ion channels in brain cells to stop or reduce the seizures. And we can also use anatomical techniques to try and understand more about, are there discrete populations of brain cells that are critically important for this seizure-like activity. And really the most important thing that we're trying to do is we're trying to use these techniques to test ideas, hypotheses, about how we can stop or reduce this aberrant electrical activity, this seizure activity. So I'm now going to give you an example of some of that work that we've done and tell you a little bit about why we believe it's important. So this was a person, this is a brain scan, an MRI brain scan, and you can see the white arrow is pointing to this discrete part of the brain. And this is a type of cortical malformation. So it's a small piece of tissue that hasn't really developed in the proper way that it should. And because of that, it's causing very difficult to treat seizures. So this is a focal cortical dysplasia. And it's a particular type which is very, very hard to treat. And this person underwent surgery, and we were able to obtain the brain tissue, again with consent, from this patient, and we were able to take it back to the lab. And then what we can do is we can use these very sophisticated arrays. These are called microelectrode arrays. So these are very tiny little silicon probes, less than 100 microns, so just think about that. They're very, very small. And on each one of these silicon probes, you can see that there are tiny little contacts. And these are little electrodes that we use to pick up the activity of the brain cells that are in that piece of tissue that we are keeping alive, okay? And then what we can do is you can see here that this tissue starts to generate these seizure-like events in the tissue that we kept alive in a bath, okay? And what we can then do is we can pull out the behavior of individual types of brain cells, and we can monitor and record how they behave as the slice, the human brain slice, epileptic brain slice, is generating this seizure-like activity. So this is a fantastic platform because what it allows us to do is then test specific ideas or hypotheses about how we might stop or reduce the incidence of these seizure-like events. And I'm going to just show you an example of some of the data that we've obtained from these types of studies. This is an experiment where what we've done here is, this is the control, so this is the baseline, and you can see that the slice is generating these seizure-like events. And then when we perfuse, when we add a particular type of drug, this drug is called perampanel or Fycompa, so it blocks a certain type of receptor in the brain, you can see that this activity is completely abolished. Moreover, when we look at the activity of neurons, the neurons which underlie this network activity, we're recording from hundreds of neurons at the same time. You can see that every time that you have this seizure-like event, the neurons all fire together. So they coordinate and they generate the synchronized activity which gives rise to the seizure-like event that we see here in the network. And when we add perampanel, what we see is that that burst-like behavior gets abolished. And this is very important because it tells us novel information about how this particular receptor is contributing to the seizure-like activity. So that's all very well and good, and it's an interesting piece of science. But what are the implications as we go from what we do in the lab to the bench, from the bench side to the bedside for the patient? And an important part of what I think is critical for epilepsy research, and which is obviously occurring through Future Neuro, is translating to clinical care, to patient care. So at the same time as we were doing this work, a colleague of mine in Newcastle, Dr. Ramesh, who's a pediatric neurologist, he had a young boy who had a very similar type of malformation. And what I've just represented here is basically a seizure diary. So this is the number of seizures, and you can see that throughout December, things are starting to ramp up. And various anti-epileptic drugs have been given to this young boy with some success, but then things get really bad in January, and he's having nearly 30 seizures a day. So things are not looking very good. And Ramesh, in communicating with me, I told him about the research that we were doing, and we were talking about the usefulness of perampanel, and he decided that they would give this young boy perampanel as an add-on. So they started him off on a very low dose of perampanel at 0.25 milligrams, and you can see that it's clearly had a dramatic effect. There was a bit of a breakthrough series of seizures here in early February, but they increased the dose, and again, he's got fairly good seizure control with perampanel. So hopefully what I've been able to do today is give you an explanation of what neurophysiology is, about what we do in the lab, and about how I think it's important for providing better answers in terms of how we treat seizures. And again, I would just say that it's very important, I feel, in my role at Trinity to engage with the community, to engage with Epilepsy Ireland, and I look forward to hearing from you and being involved in future activities. So I'd just like to thank you for your attention, and here's my email address, and if any of you are interested, there's some further reading, don't worry, I won't be testing anybody, but other interests of mine are mitochondrial epilepsy. We just had a paper published in Brain last week on mitochondrial epilepsy, and I'm also very interested in brain tumour-related epilepsy as well. So once again, thank you to the organisers, and thank you to your attention. Thanks very much.